Okay, so even if we were meeting face to face, we would have gone to Thayer Ridge Road. And uh, that's a really big area, and a lot of it we can't go on to because it's an old landfill that's not like mature enough yet to like let people onto it. So you're constantly, when you're outdoors, which ecology is often outside, yeah, you have to find a way to count like every animal and every plant. That would be ideal. You can't. So this is a sampling technique. So the Quadrat survey, just the more quadrats you could do, the better. So like the Owl Pellet Lab, when this is also a job. This is especially as an undergrad, they'll pay armies of students to go out in the summer and go again to like woodlands, preserved areas, places where again you wouldn't be able to count every single thing. But the idea with the Quadrat is if you get a regularly sized sample and get enough of them, that data can serve as a representative of the whole. If that makes sense.
Yeah.
But this is another straight up ecology lab. Ecology has just developed a lot of techniques on the fly for doing things in the field. So that I think that's why environmental science borrows from it so much. So if we were in the lab, we would be using these like pre-measured quadrats. And they're like PVC squares. And the manual has you guys doing, I think, a 3-meter square. It doesn't really matter. The larger the quadrat, the more data, the more representative and more accurate the sample is. But you also have to balance that with, again, you can't count everything. You don't want your square to be like, a square acre. That'll take you a minute. This gets, like, we're just going to do the one square. If you wanted to, like, even if I wanted to do my whole yard, I would make like probably a checkerboard of squares, because again, I know I can't do I can't cover everything, but there are different sampling techniques for different purposes and different ecosystems, different shaped areas. Like mine's pretty regular because we're out on 229 a fair bit, pretend country. Used to be country. So it's pretty square, but you don't, when you're in a forest preserve, obviously you have these really irregularly shaped areas. And Thayer Ridge, we would have an irregularly shaped area where we would even be allowed, and then again, like we wouldn't even be allowed to go on the landfill part. So anyway, I'm gonna suggest, since you guys are at home and we don't have meter sticks, if you have a meter stick, terrific. Go ahead and measure out 3 meters. If you only have a ruler in feet, it's about 3.3, I guess, if you want to round up, 3.28. In the field, another thing ecology is really famous for is it's like one of the only science labs you'll get a job in because you know how to use a paperclip, like 35 different ways. And then environmental science came along and like standardized, and now we have things like pre-built quadrats. But back in the day, or as an ecologist, if you run out of quadrats, a lot of times somebody forgets to pack it, and you leave for the field site, which might be hours away, and you get there and you don't have your equipment. So you make stuff up. And so I'm going to show you I have some ideas on how you can make this stuff up at home. And I have some yarn, And then for the length, I'm just gonna, go hand to hand. I'm just gonna do my arm span, because then I know I can measure that later. Like if anyone in the house has a measuring tape, or again, if you have a ruler, you can put it down and get that measurement later. And if I'm using scissors, but if I were out in the field, I would probably use my pocket knife. And that's just as a field worker, you get a pocket knife and you get cargo pants for all the weird stuff ecologists need. And you just make 4 of these. If you don't have yarn, just find anything that you can have 4 of, or at least 2 of, so that you can make it up outside. Could be really anything. I went out in the garage and let's see, I think this was an old rack. I just found like random things, but all these guys are regular, so I could use 4 of these. And then worst case scenario, this guy's a little small, but you could take a box and just, it doesn't have to be square for us. It doesn't have to be perfectly square. The squaring purpose is, again, if you were doing a really large area and you want all of your samples to fit together nicely. But something like this would be close enough. I would do 4 of these though, to be equivalent at least to the type of size you would want to sample for something like this. If I took that out in the backyard, I probably wouldn't find anything. It's a little small. Okay, so I'm just gonna get 4 of these. You guys can use anything. Get dental floss, like raid that weird drawer in the kitchen if you have that. I think that's a normal thing to have. There are strings, if you have pets, on those food bags on the top. It gets creative. And believe me, these are like job skills when you get to the ecology lab, to be able to find things like this. Okay. I already filled out my data sheet. So you can do that, especially because it's a little cool outside right now. When you do field work, you try to minimize particularly pen and paper stuff before you go outside, if you can. So, I went back to that analysis, page 91. And I've already got, stuff I could fill out before I go outside. I don't know why it doesn't have a date. You just don't ever not date stuff in your life, like ever. Like whatever it is, whatever notes you're taking. So I put the date, type of community being studied. I put backyard. That's a little weird. Again, we would have put, we probably would have put like natural area, park. We could have put former landfill, something like that. What else? You can identify like woodlands. I guess my backyard is really like a grassland. I don't know, that seems like a stretch to call it that because there's so much human disturbance, but that's the kind of thing they're looking for. So if you do this like in your backyard, or if you want to go to Fair Ridge or any other park in the county, you know, you can put down what you're doing. The Quadrat area, again, because if you were in the field and you just hadn't measured the arm span yet, I just put arm span square for now. And then again, when I'm done with this, I can come back in out of the chilly air, and I can put those those yarn lengths down, and I can measure them. You know, even if you only have a little something little, you know. And then let's see, I'm going to write down the weather for the initial observations of the community. I broke it down into the weather. So I'm just going to look up some basics, like the temp. 35. Hello, spring. 35. Conditions, you know, you put cloudy. It says cloudy. I'll put partly. It's pretty sunny. Let's see what else. I broke this down into vegetation. And animals. And then, you know, I wrote down, for me, it's mostly grass. We've got some sparse trees, but it's mostly like exposed. There's a couple garden areas. But again, you'll do this wherever you do it, so you can describe it with broad strokes. And then for animals, I put what I could see now, because my Quadrat's not going to include a lot of the activity, things like birds and squirrels. There are a couple of hawks flying around today. Yes, I kind of even wrote it down. The birds I broke down into the songbirds, mostly because I put some peanuts out there to hedge my bets, because I didn't want it to be boring. And the predator birds, the birds of prey, squirrels. There's some dogs. A couple of my neighbors have their dogs outside this morning. There was a human, technically. So you have a question about human disturbance. So don't forget to include, you know, human stuff. But anyway, this is like the broad strokes for just the whole area of what you're doing. Also, like knowing how many humans or like dogs or birds, particularly insectivore birds like the robins who, you know, hop on the ground, that'll mean that's important to know later, because if you see a lot of worms or don't see a lot of worms, or things like maybe like potato bugs, or that can, knowing that there were a lot of insectivorous birds, you know, on the bigger level can, you know, help put your Quadrat, you know, it's the difference between, you know, zooming out and zooming in. One of your homeworks recently was asking you why you want the bigger picture. So a lot of you guys got it. Identifying that it's a different level of information. It's more information, but you also, you know, we're going to balance it out with the minutia. So I think that's all I did. I made another note that it was pretty quiet. Things like worms are really sensitive to like footsteps. So believe it or not. So I made a note that it's pretty quiet. And then I was going to point out some things. We're going to use 2 data sheets. It looks like they gave you one for plants and one for animals. So I'm going to include insects on the animal types. data table, page 93.
So is this all surface or do we dig?
You can dig. I wasn't going to require you because I don't know what people have at home, but we would have taken a trowel really carefully though, because you want to be able to like make a divot check and then put it back, because we would have been at a park, and you cannot be digging holes in county parks, obviously, but it's your backyard. So if you have a trowel, the last question, nope. One of the questions I think has you dig. Maybe the last step. Yeah, the last step on like the methods on page 90. It says use the trowel to take several large samples. They're talking about in your Quadrat, and then if they would have you sort it the exact same way you did for the biotic components of soil lab.
Okay.
You can do it, but they're yeah. I was gonna make that optional. Just again, because I don't know. I don't want to assume everybody has a place to dig. My parents would have been a little weirded out by that. A little. So we were like in town with a pretty small lot. And, you know, I just don't want to assume we can dig. But good question. This is mostly O horizon, A horizon. Here's another thing about sampling, just in general. This is a completely impossible goal, but the ideal is to minimize your presence. So you don't want to kill things on your way to counting things. And you don't want to be disruptive. You want to see what's out there, and your presence is, it's you can't minimize it completely. You can't like negate it.
Yeah, my backyard would be pretty obvious that I've been there plenty of times.
But also like counting things, like just like I said, like your footsteps are going to affect the insects, the very insects you're trying to. I mean, it's hard to not disrupt the very thing you're trying to observe. So this really is like the skills you learn like in scouts and hunting, and right, like you're trying to be there but not let the animals know that you were there. For the plants,
right,
it's more like we also be very respectful. So the other thing that's going to go on out there, they recommend, so there's going to be some identification that you do on the fly. Oh, so maybe I should tell you that I use apps now for field guides. You almost have to take an entire class on field guides to know how to use them if you're not familiar. So I wouldn't really worry about it too much. And again, you're not gonna, like my backyard, there's not gonna be too much excitement. Like it's not gonna be too hard to ID stuff. But when I don't know something, like sometimes, and also because of the time of year, it's like I have to relearn this every year. Some of the weeds out there, like whether it's creeping Charlie or henbit, you know, I'll get confused. You can always take photos now because it's just 2020. Back in the day, we would have collected samples, so you can do that too, and you can take a leaf and like tape it to your data table, you know, if you want to ID it later or collect it somehow. But we, you know, again, we're trying to minimize our disruptions. So you be kind. You be like, there's ethical ways to sample. You don't go out and pull the whole plant, kill it. You don't take off so many leaves that you effectively leave the plant without photosynthesis capabilities. you take whatever little bit that you need, and now photos will go a long way. Plus, you can download apps, and there's a lot out there For plants, I like the one called PlantNet. That's about the best I've seen. Most of them are just not great. And most of them will give you so many options once you upload the photo that you kind of already have to know a little bit about plants. But I think PlantNet, um, that's the one I go with. And there is actually an app for tracking animals. So obviously, there's no way that I'm gonna put my little square yarn down and get a raccoon, but I know they exist out there, and I'm not gonna get a squirrel either. And I can see one right now, but he's gonna scurry away.
Yeah.
But sometimes you see the indirect evidence of the animals, and they list a few on page 90 for your methods. And so again, if we were at Thayer Ridge Road, we might see something like scat, and there is absolutely an app for identifying scat. It's last I had it was just really big, so I think I took it off my phone. But if I were about to go out in a real, not just my backyard, I would probably like go ahead and put it on my phone for the day or whatever. But it was pretty big file. Yeah, app. So there's all kinds of IDing apps. Because that is part of the process. If we were at Thayer Ridge Road, we'd talk about mostly what you're going to see. It's a lot of grass, but they have a lot of moss, a lot of the other like non-vascular plants. So you can even download like apps specific to bryophytes, which is like the scientific category for moss. Okay, so I'm just like looking at these. I'm going to even them up. For some reason, one's really short. I was probably talking. Okay, and then I'll take that outside. Just going to double check. They recommend taking like a bag. I'm not going to bother because that's for collecting specimens. And I would just take a picture. Unless I saw something cool, like if you're going to see a 4-leaf clover, this would be the time of year probably, because especially since you're going to be zooming in. You're supposed to take if you need a magnifying glass. I don't find that it's always necessary, but I don't have one on hand, so I'm just going to use my phone, and I turn on the camera and just zoom in. I have a macro lens I could put on too, like for my phone, but I'm not going to put it on because I have to take it out of the outdoor case, and I'm famous for destroying phones when I go outside. At least I'm not near water. I thought that was going to happen for the stream monitoring lab, but it did.
Oh yeah, I can see that.
See, a pair of forceps. If you're going to dig, you would take a trowel. They also recommend taking a tub or a dish pan, and that's for when you probably, if you're sorting things out, particularly if you're digging and you want to do the soil biota. They say plastic gloves, but I mean, you don't go somewhere where I don't know. I'm not going to wear gloves because it's my backyard. I'm not worried about anything. So somewhere grosses you out. Don't go into a super fun site and do this. Stay somewhere clean. Insect net field guides index card. Again, I'm just, when I go outside and do field work, you want less is more. I'm not going to use an insect net or anything. Okay, I'm just looking over the questions. The number 2 asks you about expectations. So you could take a minute before you go out and kind of, you don't have to write a hypothesis or anything, but you could kind of generally put what kind of levels of biodiversity, more or less. I mean, you could just circle more or less, because I mean, I'm not expecting a lot just because that 35 degrees. Animals are like us. That's a little cool. Also, again, I put the peanuts out there for the songbirds. So they're probably like listening to the birds and hiding. I'm gonna circle less. Other plants, specific surrounding areas. If you fill out page 91 with the initial observations, that's pretty much the answer to number 4. So you already kind of do that work for yourself. And number 5 is a little off because, again, we would be going to a repurposed landfill that's now a park for human use. So that's kind of an optional question. You may not have like a recommendation for your backyard, or maybe you would. Maybe you'll see that like the grass needs aerated or mowed better, or some kind of something. Maybe you think you need more plants because you want to see more biodiversity or something. So you would take that out in the field, pack that up, and then I'm going to get my phone hooked up so we can switch to that view outside. Take a last sip of coffee. I'm going to head out. Okay. If you had a pre-measured quadrat, you can just throw it, and wherever it lands, that's kind of an unbiased way to start your sampling. I don't have the pre-measured quadrat, but I can just toss, like one side. And then line the others up, kind of off that one. Because you don't want to walk out and you don't want to like pick your area. You're trying to get a representative sample as is. You don't want to say, oh, I want that spot because it has more or less. You gotta be, like right now I want to include this little plant, but I had to be sure it was technically in the bounds. I'm gonna set up the view from my camera so you can get a overhead. Can you see that? That's close to something like square meter. Straighten that up. And from here, you just you sample, you count the plants, you count the animal evidence. Already I can see dog scat. Let's see. So you want to be kind of even fairly regular within your square. So want to break it down into quadrants. I should have brought that box out. You really zoom in. Let's see. We've grass. It's probably wild violet. This is probably a dandelion or dandelion species. Here's evidence of a plant that's not going to be in my quadrat. That's actually a tree flower. So you might find things like that. If there are maple, I don't think it's too early for them to have deposited, but from fall last year, you might see evidence of like little maple helicopter seeds. So you count that if it's in your quadrat. This is the same tree flower, but it's kind of like dried up here. And because you're counting, when you're listing the species, you don't have to list every single grass plant. You just, grass, you just count it once. But at some point, you have to kind of get an estimate of the percentage. So you use that square space on that first page to sort of map out. So, I would use different shadings. Like for grass, this is predominantly grass, so you could either like visually show that most of it's grass by like doing a light, shading in the square and then making a note that's mostly grass. And, and then you can count the rarer ones, the random violet, dandelion. I've got some leaves here. Between this and the flower, I know there's a cottonwood tree. I just know that because I know that. You might want to get a picture. This is another tree part. Somebody lost their branch tip. I don't know what that one is. I would have to maybe collect that one, get a photo. Here's some thistle. Here's some more violets flowering. But you could tally those. Just keep like tally marks as you see them. And I'll just at least give you an idea of who's out there more or less. If I move over here, this is a species, I think it's called Muscari. You might see something like that. I see some dandelion flowering, but they're not inside the quadrat. But I could use those to double check these leaves. Dandelions. And that's really all you do. You want just a head count of what species are here, and then an estimate. If I only see 1, 2, 3, maybe 4 or 5 violets, I know that's a really low percentage compared to the grass. You could also kind of, again, if you break this down into smaller squares, I mean, you can really do that percentage a little bit easier. But you don't have to do a percentage. I would probably say 99% grass and then break that last 1% up between the violets, the dandelion, the thistle. I'm not seeing too much more. You might see some clover while you're out there. Oh, there's some clover right here. This looks like it's going to be red clover. So for animals, you do the same. Got dog for mine, and you kind of have to get between the plants and see if you can find anything. Usually you set a timer for this. And, 15, 20 minutes, that's a good waiting game. And again, you can wait. You don't have to do this at 8 a.m., 8:39 a.m. on a 35 degree. You could do this today in the afternoon. The warmer it is, the more activity insect-wise you'll find. And if we were to dig, we would probably find plenty because they go down when it gets cooler. If you really want, there's a sampling technique where you can mix mustard with water and pour it on the ground, and a lot of the insects will come up. You can email me. I'll send you that protocol if you're really interested. It'll bring up the night crawlers for sure. And I don't know, that's really all there is to it. Oh, now that I'm sitting here, I can see more like moss stuff. This stuff. The longer you sit, I would say sit with your first sampling, like when you break down your Quadrat. Sit with that one probably the longest because your eyes have to adjust to all these shades of green. And if green's a difficult color for you to see, this is tough. You're looking for like different plant textures. For insects, movement, but it's slow movement. But so you kind of have to sit there. Might take 30 seconds, to let your eyes focus. To be the cat. Be like the hunting cat. Watch for blades of grass moving, not with the wind. That might be something crawling away from you, probably, because you're poking. And you can bring forceps, so you can like pull back plant areas finely. I just don't bother. It's like more violet. Yeah, anyway. But yeah, I would definitely cut myself off. I wouldn't do the whole thing for more than 30 minutes. Because you get kind of in that weird place where you want to find more stuff, and it's just not a great time of year. I mean, don't sit there forever. You wouldn't do that. In the field, you would just have an agreed upon time, and it's usually around 20, 30 minutes for a sampling. And then you want to get up and you want to move to a new area. And as long as everything stays the same amount of time, that's a good, remember you're sampling. You're not actually taking a census. So don't make it hard on yourself. Any questions? Before I'm gonna run back in because my hands are freezing. I should have brought out I've got gloves with no fingertips.
Yeah, I did this very spectator-like. I made another cup of coffee and watched you go outside.
It is warm. It looks warmer than it is, that's for sure. Whew. My eyes are watering too. Light. Anything you want me to re-go over or summarize?
How did the exam with your other class go over?
Oh, let me grab my coffee.
Definitely.
Good question. Here, my phone in case we need that. It was fine. I only had a handful take it with me, like during our regularly scheduled lecture. Most of them have made appointments because a lot of them have a full house. And got a lot of we had to wait for some young bedtimes last night.
Yeah.
So that was no big deal because I can either, if I'm up, I can turn on my Zoom. I mean, it's not really a big deal. It's almost like I felt like, so they would turn on, they would log on to the Zoom and at least have just the camera on them. We didn't have any technical difficulties with Moodle. I found that most people were like faster than usual, faster than the pen and paper Scantron. Maybe just because we're not transposing answers.
So it's like the quiz is multiple choice. You page through it. Okay, that's how my other exams have been with my other classes, but they're all open book, open notes. So that's why, for you to enforce that doesn't happen, is why we're doing this through video. Is that correct?
Right. So that I don't have to make it essay.
Right.
I want to keep it the same format as the past exams.
Yeah, I get
it. So what most, I guess, well, I only had like the three on at the same time. But whether you do it during our regular lecture time or on your own, you just use the share screen button at the bottom, and then I can see I can see the screen, and I just kind of sit here. It's almost like a virtual study hall, or I guess the same way it would be in the classroom, really. But it was just kind of nice to be here for technical, in case anything went wrong. I was here for when everybody submitted it, which was nice. So I could verify that, like, yeah, I can see it. Like, you're all good to go. And the other difference was if some people who did it on their own, who did it really late last night, and I couldn't Zoom with them, they hit the record button and did the exact same thing. Just shared the screen.
Is there a Dropbox you put it in, or with exams, are they automatically submitted when you're with them?
Once you finish, I think there's one click to submit it, and
okay.
And it just, I don't know, it went pretty uneventfully, I guess. Knock on wood.
Yeah. Well, right now I plan on doing it Thursday during class time as scheduled.
Yeah. So that exam was like 50 questions for them. This was microbiology, so it was a lot of matching for them. And what used to take maybe 40 minutes seemed like it was taking 30 or less. I think the first person finished in like 25 minutes. So 50 questions, and I thought it felt like a little speedier.
Yeah, I don't feel like I would go back and review as much with on screen. So I don't know if I'd be more terrified of my grade or not, but I can see why I might maybe take it less seriously. I don't know. I'll have to see.
People are doing well. Someone hit a wrong button too, and I was able to like fix that in real time because it was almost like an oral exam. We were able to talk it out, and that was kind of nice. Like, because I can literally be on the other side of Moodle and watch you answer. I have to refresh the page, but I mean, I can. It was kind of nice. I felt like that was really helpful.
Yeah, I think it would be.
I don't know. Seemed okay. I have one last weird issue to tackle, I think, this semester, and I think everything like, you know, our labs have really helped me figure out how to do this on Friday mornings, and we've knocked out a lot of lectures. I don't know. Now the exams, I can see the light at the end of the tunnel, which is good because the calendar, the time between the last day of class and today, it's like really small.
Yeah, I'm starting to feel that too. After an extended spring break that I think most is what it is.
Yeah.
So,
Any
all
other
right.
questions?
No, I think that's pretty good today.
Yeah. All
It'll
right.
be review Tuesday and the exam Thursday. Is that correct?
Mm-hmm. Yeah.
Sounds good.
All right.
See you
Well,
later.
yeah.
Have a good weekend.
You too.
Bye.
Bye. Okay, guys, it's about 9:33. So I'm going to go ahead and log off. I don't know that anyone else is going to join in, but along the way, as you do this yourself, if you have any questions, email me. I did forget to do one thing when we were out in the backyard there. If you have a Frisbee, that can be useful sampling. You can use that in a couple ways. You can use this to figure out where you want to sample. Remember how when I was out there, I kind of threw one side of my Quadrat so that I did an unbiased location. Actually use the Frisbee to do that for you, and then you can do your square wherever that Frisbee lands. And then if you want to sample more than one location, you know, if you're in your yard, or wherever you go, if you just want to do more than one Quadrat, the Frisbee is a really unbiased way to decide where to put those. So just wanted to let you know about that. Otherwise, I'll see you guys next week. And Tuesday, we're going to review these Amazon chapters 15 through 21. So we have water resources, atmosphere, climate change, and then the fuel sources, the fossil fuels, and then the renewables broken down between conventional and new. Thursday is the exam. There's some information on Moodle. And then if you watch this recording, Darren and I talked. He had some questions. And otherwise, you can email me, and I will see you guys next week. Happy sampling.